right? Protein metabolism, right? So normally what we eat contain basic nutrient. What are them? Protein, lipids, carbohydrates, vitamins. So then you get another ions. So whatever in toxin, so lot of things. So then, so you need to know about glucose metabolism, carbohydrate metabolism, and the protein metabolism. Right. So now here, if you summarize the protein metabolism, the summary that has been given here. So now catabolism. What is catabolism? So catabolism means, so you know there's reactions in the human body, right? Anabolic reaction and catabolic reaction, right? Anabolic reactions are building up reactions, while the catabolic reactions are breaking down reactions. Catabolic reactions are breaking down reactions. For example, building up of proteins from amino acids are anabolic reactions. Right. So now what you have to know that here protein catabolism, where it occur. So you know that. So location, stomach, small intestine. So step one, so it happened liver, kidney, small intestine. And step two, liver and kidney. Right. So that, you can see now here, there's arrows in either side, so that we'll discuss about it why. So then, digestion and absorption of dietary proteins. There's a two type of proteins, so animal proteins and plant proteins. Right, so that it has been said that human need animal protein as well, because certain amino acids only you can have it from animals. Right. So digestion and the protein starting. So where the protein digestion take place, the carbohydrate digestion start while they are in our mouth, right? So where protein catabolism start, protein catabolism start.
At the stomach?
Yes, yes. Catabolism start at stomach, right? Right? Because carbohydrate catabolism start from mouth itself, because that our mouth contain enzyme ptyalin. So ptyalin act on the cooked carbohydrates. Right. So now here, protein made up of amino acids. Right. So we are going to talk in a minute about the structure of the amino acid. So now it's protein catabolism starts at stomach and it continues along the small intestine. So protein are complex molecules, and every protein made up of amino acids. So you get a primary structure, secondary structure, and tertiary structure of a protein. So the tertiary and quaternary structure. So that means the primary structure is the simplest, and you get amino acid sequence. But when it comes to secondary and tertiary structures are more complexes, right? So when amino acid, so digest start from the stomach and it go towards the small intestine. So amino acid, then there's a two process, transmination and oxidative deamination, right? So there's a two ways. That means it go amino, ammonia and urea cycle. And other way it go for the carbon skeletons. So under influence of aminotransferase, glutamate and dehydrogenase. So you get metabolic intermediates. These are oxaloacetate, pyruvate, alpha-ketoglutarate, fumarate, succinyl coenzyme A and acetyl coenzyme A, and acetoacetate. Right. So now we are going to talk about more information about the removal of nitrogen. Now this diagram, This is an amino acid, structure of the amino acid. So it's it has centrally situated carbon atoms, and it has four groups. You can see these groups are not equal. Right. So amino acid NH2 group, and here COOH group is a carbonyl group. So amino acid group, so this has a basic properties. And this one, carboxylic acid group, which has acidic property, this is unique heat, right? So remove alpha amino goes. So essential for producing energy from any amino acid, obligatory step of amino acid catabolic. So that you have to alpha amino group. So here two step. Transamination and oxidative deamination. So that means removal of nitrogen happens. So nitrogen only here, no? Right. Removal of nitrogen happen in two steps: transamination and oxidative deamination. So end products are right. So end products are alpha keto acids, right? Alpha keto acids. Then it entered the central path of energy metabolism, ammonia and urea. This pathway is essential, and also that you will learn about some illnesses which prevent amino acid catabolism, right? So now here we have to know about the first step, right? Transamination. Transamination. We'll see what is transamination, right? So that remember that when you are learning biochemistry, right? You have to remember a lot of things. So easiest way to remember, so you have to read them over and over again. So when you do it over and over again, so it naturally go to our head. Therefore, don't worry, right? So therefore, so that if they ask about definition of what is transamination, it's a reaction that transfer amino group from one molecule to another, typically from amino acid to keto acids, right? This process important in amino acid synthesis and degradation. So that means a synthesis and degradation and catalyzed by enzyme called transaminase or aminotransferase. So that if you look at this one, so key players here are alpha ketoglutarate that accept amino group. Amino group is this amine group. Amino group from most amino acid, the glutamate and alpha keto glutamate and alpha keto acids. So that you can see now here that alpha amino acid here, that there's a small diagram. I think you can. So I'll enlarge the diagram. Then you will see it is. Right. So that you can see now here. This is alpha amino acid. This is alpha ketoglutarate. Right. Amino transferase, right? That is alpha keto acid glutamate, right? So you can see that the arrow, it is in a two-way. So that means the reaction is reversible, right? So now here, glutamate, right? So this is the glutamate. 8. So 8 means it has, right, negative charge. 8. Right. Can be oxidatively deaminate in step 2. So we have learned about the step 2, right? Okay, so because these notes are very organized, whatever, this note, right? Or act as amino group donor in the synthesis of non-essential amino acid. So now we have to know there are two type of amino acids, right? These are essential and, right, essential and non-essential, right, amino acids. So essential amino acid, there are 9 essential amino acids: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Right? So that means about 9. So there are, right, non-essential amino acids. So non-essential amino acid, so there are 11 non-essential amino acid: alanine, arginine, asparagine, right? Aspartic acid, cysteine. So you don't have to remember those. So what you have to know, there are two types, essential and non-essential amino acid. So there is another one you have to know, amino transferases. Amino transferases. So that if they ask about that one, what is aminotransferases? So aminotransferases known as enzymes. That means a group of enzymes, right? That catalyze the transfer of amino groups between amino acid and oxo acids. Amino acids and oxo acids. Okay, please write it down. What is oxo acids? Oxo acid. Oxo acids. So oxo acids are what? Right? Oxo acid, sometimes we call oxyacid also, that contain oxygen along with at least one other element and at least one hydrogen atom bounded to oxygen. So oxo acid, you have to know about the definitions of oxo acid, right? Oxo acid, and they are, they are essentially in our metabolic pathways, oxo acids, right? Amino acid, right? Aminotransferases. Found in cystole and mitochondria. Sorry, cytosol and mitochondria. So you know that mitochondria we have learned, right? Mitochondria we have learned. Inside the mitochondria, you get this aminotransferases, right? So those are they come for the MCQs, right? You have to know now, right? Cytosols. Cytosol is the cytoplasm, no? Cytosol is the cytoplasm, right? Normally we call cytosol is the fluid part of the cytoplasm. All amino, so then amino transfer, especially in liver, kidney, intestine, and muscles action. Transfer of alpha amino group to ketoglutarate. So don't worry, the ketoglutarate come later, right? All amino acid undergo transformation except lysine and threonine, or threonine, lose their alpha amino group by deamination. So you have to know about, right? Again, transmination. Transmission, because over and over again you have to remember that. So transmination, right? So transmination we discuss in a while. So what does it mean, right? So transmission is a chemical reaction that transfer amino group from one molecule to another. So amino acid to keto acid. So that is transmination. Except lysine and T threonine, threonine, threonine. Right. Next one. So that we are going to discuss this major pathway. So major pathway. So it is transmination. That means amino transferases. Each amino transferase is specific for one, at most few amino groups donor. Named after a specific amino group donor, all require coenzyme pyridoxal phosphate. Some pronounce as a pyridoxal or pyridoxal. So it's a vitamin B6 derivative. So that's what we say, that you need vitamins for this, right? So in here they have added some additional information. So what is this additional information? Right. So uh, liver function test, ALT, right? ALT. Right, so that these things are important, that we say if it in liver function test you get ALT, right? What is ALT? The significance of ALT, right? Significance of ALT. ALT means alanine aminotransferase, right? So it is, what is that? It present in many tissues, catalyze transfer of amino group of alanine to alpha ketoglutarate, so pyruvate and glutamate. So it is a reversible. So the arrow to other side is the reversible reaction. So that means a alanine transferase. ALT, lot of people that confuse ALT and AST, right? Alanine aminotransferase, right? So it is a enzyme, right? So we use that. So there's a code in blood test, liver function test, you get ALT. So if you're a clinical biochemist, you will know that, right? So ALT test, right? So there's a ALT test means that means we used to identify liver problems. For that means elevated ALT level can see early indicator of liver damage, right? Elevated ALT, right? So then normal ALT suggest that means you are a healthy person, and very high ALT is a liver tissue, that liver damage, right? And also that you get the moderate high ALT, moderate high ALT. So it is associated with alcohol consumption, and also in the obese people that you get it, you get a very high ALT, right? Very high ALT, very high ALT. Uh, so you get a hepatitis, that means a viral infection, right? Hepatitis, right? So now here, there's another name. Other names for the ALT, you have to know that. So that sometimes it get SGPT, serum glutamic pyruvic transferase, or GPT. So now we use the ALT. So you look at the ALT now, you get a pyruvate, right? Alanine. So glutamate, alpha ketoglutarate. So alanine transferase is what? It is what? Alanine transferase is what? It's an enzyme, no? It is enzyme. ACE, carbohydrates. Next one is the AST, aspartate aminotransferase, AST, right? So AST transfer amino group from glutamate to oxaloacetate to aspartate and vice versa. Important urea cycle, this is reversible reaction, right? So it is enzyme. So you get in liver, heart, muscles, right? Liver, heart, muscles, right? Notably, and other muscles also there, right? So normal range. So normally 8 to 33 units per liter, right? So abnormal results. So elevated AST can indicate liver damage, so muscle injury, heart attack, so that we can discuss these things later. So you have to know now here. So a function of this ALT and the AST, right? Next part. Keto acids. In biochemistry, what happens? So you need to remember this, but it doesn't matter whether you remember or not, because why? You have to read this over and over again. Sometimes what happens? The key parts, you have to, remember. That means you must be able to draw structures, right? So that means learning medicine is. So because why then they want you to remember these things? Because when the patient comes, yeah, obviously you can search on Google and find what it is. But the doctor who lived 20 years ago, you didn't have AI. So all based on memory, right? So therefore that this is the right? So you can't just wild guess because the patient dies, right? So therefore you have to remember a lot of things. And in biochemistry, so you can't talk about medicine without knowing the biochemistry. So biochemistry, physiology, because these all are interrelated, right? So you can't isolate at all. So we can't isolate these things at all because each and everyone is related. Each and everyone is related. So you can't isolate. Right. So now we'll see about the keto acids. Keto acids. Right. So it's a very low carbohydrate. Right. So that means a we call keto acid. It's a very short carbohydrate. That means organic acid. Keto acid is organic acid that contain both ketone group and carboxylic acid group. Right? So that previously what I said about the keto diet. So if you ask about the keto diet, means an organic acid that contain ketone group. So you know that ketone group means, so there's a two groups, aldehyde and ketone. Right? So keto. When you say ketone, actually, it's a functional group of carbohydrate. That's what I said before. It is a carbohydrate. So keto acid, and this one, it has, right, carboxylic acid group as well. Right? So that means a keto part and the carboxylic acid. So they have colored it beautifully. So ketone group, if I draw it, ketone group, so this is the ketone group. Right? Carboxylic acid group, so you get, right, this, no, carboxylic acid group, OH. Right? So when you say about the keto group, and the carboxylic acid, so that's what they have highlighted here. So keto and the COO group. Right. So now here when you talk about, so the keto acids. Right. So we are going to talk in a while. That means you don't have to remember this each and every structure, but you must be able to identify at least what is keto acids. Right. What is keto acids? Right. So this one is the alpha ketoglutarate, succinyl coenzyme A, oxaloacetate, pyruvate. So we remember these things, right? Pyruvate is what respiratory substance. So after glycolysis, the product is the pyruvate, right? So what is alpha ketoglutarate, right? Alpha ketoglutarate. Alpha ketoglutarate. Alpha ketoglutarate. So what is it? So we call AKG. It is involved in the so many metabolic pathways. So it is important in the energy production. Alpha ketoglutarate, right? So you have to know about what is it actually. It's a intermediate. That means it's an intermediate, right? Intermediate of the TCA cycle. TCA cycle means tricarboxylic acid cycle. Alpha ketoglutarate, succinyl coenzyme. So these are the functional groups, functional groups. So they have highlighted this is a functional group, right? So now, so those that means acetoacetate. So you don't have to remember, but here this one aminotransferate. So here alpha keto acid becoming alpha amino acid, and also glutamate become alpha ketoglutarate. So enzyme is aminotransferase. So you have to know about what are the function of the aminotransferase. So aminotransferase, so that we have discussed, I think, before. See, aminotransferase are found in the cytosol and mitochondria. So cytosol is what? Cytosol is, so if you take about the cell, so this is the nucleus. So this area is the cytosol. So everything we call is a protoplasm, right? So sometimes this area we call cytoplasm, right? It's a liquid part, right? So now here keto acids we discuss oxidative deamination. Oxidative deamination, right? So what is oxidative deamination? So deamination term alone, right? So deamination. Deamination means something, it is a catabolic. It's an English term. It's not a, even without looking in biochemical aspects, so we can say don't deaminate yourself. That means it's breaking down. The oxidation deamination is a process where amino group is removed. Amino group is removed. So I'm making this slide easier. That means amino group is removed from amino acid, and resulting amino acid converted to the keto acids. Right? Resulting is the keto acids. So end product, see, ammonia and the alpha ketoglutarate. So key place glutamic dehydrogenase results in the liberation of the amino acid as free ammonia. For primary liver and kidney, reversible coenzymes, NAD, nicotinamide, what is NAD? Nicotinamide. This is O level. NAD. Nicotinamide, adenine, dinucleotide. No, nicotinamide, adenine, dinucleotide. Right? Right. So this one, NADP H2. NADPH means reduce. When this molecule reduce, we call reduce amination. So this one, GPT inhibitor, guanosine triphosphate is inhibitor, adenosine diphosphate activated, glutamate, product of transamination, right? Only amino acid undergo rapid oxidation. So end product is what? Ammonia and alpha ketoglutarate, right? So what is the important here now? What you have to know? That means oxidative deamination, right? Oxidative deamination. End product is, right? End product is what? End products are ammonia and the alpha ketoglutarate, right? Ammonia and the alpha ketoglutarate, right? Okay, we'll go to the next part. Direction, okay, so if you look at this one, you can see this reaction. Glutamate, what is this? Dehydrogenase. Glutamate dehydrogenase. So what does it mean here? Glutamate converted to the alpha ketoglutarate. So you can see ammonia is releasing, right? NAD, you can see the reverse, NAD becoming NADH. So ammonia release here in one direction, right? Glutamate, alpha ketoglutarate, right? So you can see now here in other way also it can. So alpha ketoglutarate, ammonia, so glutamate. So this is reversible reaction as you can see. The reaction is reversible. So here you have to give the ammonia and here ammonia release. Right. End product free ammonia and the ammonia is NH3. Free ammonia and the alpha ketoglutarate. Free ammonia and alpha ketoglutarate. Right. Direction of reactions. Direction of reactions. Both enzymes in transamination and oxidative deamination process reversible catalyzed reaction. So depend on relative concern glutamate, alpha glutamate, ammonia, ratio of coenzyme. Disposal of aminos after ingestion of the meal contain protein, glutamic level in the liver are elevated. Synthesis of amino acid B when the levels of glutamate is low. Right. Okay, now here what happens? So disposal of amino acid. So we'll look at the diagram. What is the diagram here? Right. So even though the diagram look complicated, but it's very simple if you think like that. So transamination, right? So oxidative deamination. So then what happened here? Alpha ketoglutarate, NH2, NH3 release. And you get NADH. NADH means the reduced form of the NAD. So this is disposal of amino acids. Ammonia release. Synthesis of the amino acid, right? Synthesis of the amino acid. So what happened here? Direction is other way. So reductive amination, so transmination. So ammonia you use. So when the level of glutamate is low, this is what? This happened, glutamate level in the liver elevated. Dispose so that what happened here? Ammonia release. Transamination, oxidative deamination. So you can see, that is the one part goes to the Krebs cycle, right? NH2 group of amino acid. So you can see now here, this all enter the system. Enter. This is enter. This is exist. You can see the arrows, but see all opposite. First A and B are opposite parts, no? So you can see now here. So look at these are not a mistake, right? Alpha ketoglutarate. Look at this one. NH2 of the glutamate. See, this is NH2 of the glutamate, all in the other way around. Here you use the ammonia. Here you use the ammonia. Here you release the ammonia. So one is the disposal of amino acid. Other one is the synthesis of the amino acid. Okay, please note it down. You need to compare the later on disposal of amino acid and the synthesis of the amino acid. If you remember one thing, the other thing naturally comes. Right. Okay. Now we'll go to the next part. Transport of amino acid in the liver. So there are two mechanisms. Right. So one thing is what? It is glutamine synthesis found in most tissues combine ammonia. Actually, this 3 is not shouldn't be here. The 3 has to be in subscript. So NH3. So this 3 has to be small. This has to be NH3, right? So found in most combined ammonia. So ammonia is important, in human body. You see, with glutamate to form glutamine. Non-toxic transfer from ammonia. So ammonia is toxic. But glutamine is non-toxic. Non-toxic transfer from ammonia. In the liver, glutamine is cleavage by glutaminase to produce glutamate and free ammonia. So look at this one now. But these things sometimes, even we have done these things, you have to know, right? Alpha ketoglutarate, so ammonium glutamate dehydrogenase, so you get a glutamate ammonia. So then what happened here? So this NH4, right? So included to this CH group. So you can see. Right. So then what happened? Glutamine synthesis, ATP use. Here you use the reduced NAD. Reduced NADP means some people say NADPH2. Right. So then you get the NAD. Right. So now here, this one. So what happened here? Glutamine. Glutamine is the non-toxic transport of ammonia. Glutamine is not toxic. So excess ammonia is toxic for the human body, right? Transport of ammonia in human body. So this diagram is actually messy, but we'll there's another diagram later. We'll do that. Transport of ammonia to the liver. Transamination of pyruvate. So you have to know about. Okay, if they ask about what is transamination of ammonia, right? Transamination of ammonia, to liver. Transamination of ammonia to the liver. Right? So we're going to talk about transmission of pyruvate, then primary muscle, pyruvate to alanine. In the liver, alanine is converted to the pyruvate by transmission. So important thing. Then glucogenesis. Pyruvate converts to glucose. Pathway called glucose alanine cycle. So that we have learned even in our O-level time what happened. So protein can be catabolized to give glucose. So this is the way. Pyruvate to glucose. Right? Pyruvate, alanine. So you can see now here in the liver, right? So that means primarily in the muscle, pyruvate to alanine. In the liver, pyruvate to glucose. So this called glucose alanine cycle. Right. So now here you have to know about the glucogenic versus ketogenic amino acids. Glucogenic versus ketogenic amino acid. Glucogenic amino acid that are converted to oxaloacetate, pyruvate, alpha ketoglutarate, fumarate, and succinyl coenzyme. So provide glucose, lipid, and energy. That is called glucogenic amino acid. Other one is the ketogenic amino acid. Amino acids are converted to acetoacetate or acetyl CoA. Acetyl CoA means coenzyme may form ketone bodies, so provide lipids and energy. Right. So now here again the protein catabolism. The diagram comes. That's what I said, that there's a diagram here. Right. See now here important. right? So you have to, these things are come to the exam. What is that? What is that here? Like glucogenic amino acid and the ketogenic amino acid. You got to compare this table, right? And you have to give the examples, those things. So it is naturally you can divide the book into two. You can write it page into two columns, right? Amino acid that form oxaloacetate. We have heard these things, right? So asparagine. So then so enzyme is so what is that? Enzyme asparaginase, right? Then aminotransferase. So that means aminotransferase, that means alpha ketoglutarate converted to glutamate. So eventually you become oxaloacetate. So starting compound is asparagine, right? And end compound is oxaloacetate. So at least you have to know these two steps. Asparagine is hydrolyzed by asparaginase to form aspartate, right? So the aspartate, it has some significance. Write it down, aspartate. So it's what is the taste of aspartate? We'll talk about it. You remember at the end of the lesson to talk about aspartate, right? So it is sweet in taste. Amino acid that form alpha ketoglutarate. So glucogenic amino acids, so glutamine, proline, arginine, histidine, and glutamate forms alpha ketoglutarate via glutamate. So look at this one, glutamate, alpha ketoglutarate, right? So other one, glutamine, glutamate, right? So it is easy to remember, right? Glutamate to alpha ketoglutarate. So you use the, so here ammonia release. Ammonia release. That means if you have excess glutamate. So if you have excess ketoglutarate, so other way around happen, join with the ammonia to get glutamate. So in the next one, glutamine convert to glutamate, right? So this is one way reaction, as you can see. As the results, that means, so you get, it's a hydrolyzed reaction. You can see why hydrolyzed, water is adding, no? Right. So you get the ammonia out, right? So amino acid to form alpha ketoglutarate. So there's a chlorine, right? Chlorine, then glutamate, right? Then alpha ketoglutarate. Okay, enzyme. So it is proline oxidase and proline dehydrogenase, right? Arginine, ornithine, glutamate, semialdehyde, and keto glutarate. Enzyme arginase. Histidine, go to the urocanic acid, N4-minoglutamate and glutamate alpha ketoglutarate. What is this? Histidine, the one catabolic pathway, right? So how glutamate form from the histidine, right? So it is easy. So histidine is amino acid. So histidase, you get a urocanic acid. Urocanic acid then formed to the N4-minoglutamate, FIGLU. Then tetrahydroxyfolate, then 5-amino tetrahydrofolate. So what you have to know here, this is, they have given actually as additional information how histidine converted to the glutamate. Histidine converted to the glutamate, right? So they have given that some examples here, and they have given one complete examples at the end. Amino acid that form pyruvate. We have known this. We have known this, right? So amino acid that form pyruvate. So pyruvate is the if glucose molecules start glycolysis cycle, end product is pyruvate. Here also amino acid undergo also pyruvate. So glucogenic amino acid. So these are alanine, serine, glycine, cysteine, and tryptophan and threonine. Alanine pyruvate via transmission process, ALT. Glycine, serine, but you will wonder that, you may wonder how am I going to remember this? And don't worry, it natural process, right? Glycine become serine and pyruvate. So glycine, then it carbon dioxide release and the ammonia form. So this is all two-way reaction. Okay, serine hydroxo methyl transferase. So take a piece of paper and you have to write them. So when you write this, it naturally get, this is the amino acid that form pyruvate. So look at this one. If you look at this, see, glycine form pyruvate. So they have given one example. And cysteine become pyruvate and threonine become pyruvate, or alpha ketoglutarate, which forms succinyl coenzyme A. Coenzyme means Saha enzyme A, right? So you have to remember at least one amino acid that forms fumarate, both glucogenic and ketogenic amino. So now you have to know what is glucogenic. I discussed before glucogenic and the ketogenic amino acid. Phenylalanine and tyrosine. Hydroxylation of phenylalanine produce tyrosine. Tyrosine becomes fumarated acetoacetate enzyme phenylalanine hydroxylase. Right. So now here L. So that you know that so there's a forms. L is a one form. L-phenylalanine so become L-tyrosine. So it can go to the fumarate and acetoacetate. So tetrahydroxo biopterin, other one dihydroxo biopterin, and so these are so intermediate. Phenylalanine hydroxy is a enzyme, right? So you can see that this compound oxidized, no, tetrahydroxo biopterin oxidize. Here it oxidizes. You can say joining with the oxygen, so obviously you get water here. Right. Amino acid that form succinyl coenzyme A. This is little bit that important. So, right. So this process, right. So we discuss in coming slides, but we'll discuss methionine. So methionine eventually become methylmalonyl coenzyme A. So this is one pathway. So other one is the glucogenic amino acid. So methionine, valine, isoleucine, threonine. So this one obviously though you don't have to remember, but it's much better that. So if you have, you can see now here these amino acids. So go to the go in these two pathways, and so it eventually go with the methylmalonyl coenzyme A, and the process forms, right. Amino acid that form acetyl coenzyme A or acetoacetyl coenzyme A. So these things are the normal theory you have to know. This is important cycle. So we are going to discuss that in more detail, right? So this one, ketogenic amino acid, leucine, isoleucine, lysine, threonine, and tryptophan form acetyl coenzyme A. Then then this is acetoacetyl coenzyme. Sorry, I made a mistake. Acetoacetyl coenzyme, then acetyl coenzyme without pyruvate serving as an intermediate. This is important one. Phenylalanine and tyrosine. So it could come acetoacetate and acetoacetyl coenzyme, isoleucine, acetyl coenzyme, and succinyl coenzyme. Total 7. So you get here total 7 ketogenic amino acids. So since they have given it, so it's better to remember that all 7 ketogenic amino acids without even the chemical formula. So I don't think, since you are not doing a biochemistry degree, I don't think that they will ask about the formulas, right? Now here, so this is the right. If you look at this one, you can see now here there's a cycle goes, right? So what is so pyruvate comes. Acetyl coenzyme, citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme, succinate, fumarate, malate, oxaloacetate, right? So then what happens? So here you can get the phosphenoyl pyruvate, right? Here asparagine, aspartate, here tyrosine, phenylalanine. So these things cause them isoleucine, valine, methionine, threonine, arginine, proline, histidine, duotine, leucine, lysine. So these are the substrates, right? So what is this actually? So it's what is this actually? Glycolysis? Is it a Krebs cycle? Huh? Krebs cycle.
Yeah, Krebs cycle. There's some sort of.
Yeah, it's a Krebs cycle. Acetyl coenzyme, citrate, acetate, alpha ketoglutarate, succinate, succinate, fumarate, malate, right? Biosynthesis of non-essential amino acids, right? Okay, so now we'll little talk about the Krebs cycle, right? Hold on. Biosynthesis of non-essential amino acid. So now here 11 non-essential amino acid sources essential amino acid. So phenylalanine and methionine. Metabolic intermediate pyruvate, oxaloacetate and alpha ketoglutarate. So you have to know that sources of essential amino acids and metabolic intermediate pyruvate, oxaloacetate and alpha ketoglutarate. So that small note about synthesis about non-essential amino acid. So it's actually separate lesson synthesis of the non-essential amino acid. Right. Okay, now here biosynthesis of the alanine, aspartate and glutamate. Right. Okay, now we are going to give little exercise. Biosynthesis of alanine, aspartate and glutamate. So this is non-essential amino acid. So pyruvate, right? So become alanine. Oxaloacetate become aspartate. Alpha ketoglutarate become glutamate. So it is very simple. No? Pyruvate, alanine. Amino, right? So sorry, not amino acid here. This one. Oxaloacetate, aspartate. Alpha ketoglutarate, glutamate. Right. So these are the enzymes. So all these reactions are metabolized by, activated by aminotransferase enzyme. Right. Biosynthesis of glutamine and asparagine. Right. So this is glutamate. Glutamate, this is glutamine. So look at the basic things you have to know. Basic things you have to know. So glutamine synthesis, the enzyme, right? Enzyme, right? So you can see here now here, NH2 group has been added. NH2 group has been added. NH2 group has been added. Right. Next one. Aspartate. Aspartate become asparagine. So glutamine in this section, glutamine become glutamate. Right? So asparagine synthesis. Asparagine synthesis is the enzyme. Right. Next one you have to know biosynthesis of proline and tyrosine. Right. Proline and tyrosine. Right. So glutamate become proline. Right. Glutamate become proline. So reduction and cyclization. So see, you can see now here. So this is a linear structure basically. So this is cyclic structure. So we call it a reduction and cyclation. Phenylalanine become tyrosine, right? So phenylalanine hydroxylase, that is enzyme, right? So adding of oxygen is oxidation, right? Biosynthesis of serine and glycine, right? Serine and glycine. Serine becoming glycine. So serine hydroxymethyl transferase enzyme N5N10 methylene THF act as a carbon donor. So you can see now here 3-phosphoglycerate, 3-phosphohydroxypyruvate, 3-phosphoserine and serine. So then glycine. Right. Glycine to serine, serine hydroxymethyl transferase N5N10 methylene THF act as a carbon acceptor. So you can see now here carbon donor, carbon acceptor. Right? Biosynthesis of serine and serine to glycine, glycine to serine. Serine to glycine, glycine, so that the opposite thing of the, so you can see the arrowheads are, so here you can see here act as a carbon donor, act as a carbon acceptor. Right? But same enzyme. Biosynthesis of methionine cysteine. Right? So catabolism of right, catabolism of methionine. So, right, now hold on. Right, catabolism of the methionine, right. That means meth become SAM. So you know what is meth is methionine. SAM is what? What is SAM?
S and
S-adenosine, yeah, methionine, right. Methionine. So here, adenosine added to the meth. So if you can look at it in the left hand side, SAM act as a methyl donor. So SAM become SAH. SAH is a S-adenosyl homocysteine. Homocysteine, right? SAH. So then hydrolysis, homocysteine converted to cysteine via transsulfuration process. So this require vitamin B6 and B12, folate. So converted to methionine. So remethylation process. So you need vitamin B. That means a vitamin. If you have a vitamin B deficiency, so these reactions are affected, right? Next one is a biosynthesis of the cysteine and methionine. Right. So here also you should have slight knowledge about this one so that there's a separate video. So we'll watch that biosynthesis of the cysteine and methionine. Right. So here is the cysteine. Right. So here L-methionine. Right. Cysteine, L-methionine. So this is the, so you can see the starting compound. So you get the L-methionine. So here the starting compound, here you get the L-cysteine. Right. So that means L-homocysteine is the starting product of this both reactions, as you can see. Right. So now here this important thing is here. Here now we are going to talk about illness now. Illnesses. Right. We are going to talk about illnesses. Right. So phenylketonuria. Phenylketonuria. So that is the illness. Most common clinically encountered inborn error of amino acid metabolism. Characterized by the accumulation of the phenylalanine and deficiency of the tyrosine. Causes deficiency of the phenylalanine hydroxylase. Right. Deficiency in any of the several enzymes are required since it's BH4. Now we'll look at it. Okay. So here tyrosine synthesis. Right. So deficiencies in any of the several enzymes required to synthesize BH4, right? So deficiency of the dihydropteridine reductase or any of the enzyme BH4 synthesis lead to hyperphenyl anemia and decreased synthesis of catecholamine and serotonin, right? So this is, you can see now deficiency of the phenylalanine hydroxylase. So if you remember like that, right, phenylketonuria is characterized by accumulation of the phenylalanine and deficiency of the tyrosine. So this is important, phenylalanine hydroxylase. So this is inborn error. Inborn characteristics of the PKU. The classical PKU is an inherited disease due to 100 or more different mutation phenylalanine hydroxylase PH gene. So elevated phenylalanine, right? So phenylactate, phenylacetate, and phenylpyruvate. So, right, give urine characteristics musty or mousely odor. You get some weird smell. CNS symptoms, that means central nervous system illness. So mental retardation and fatigue to grow, failure to walk or talk, seizures, microcephaly. Okay, write down what is microcephaly. So we are going to ask it, right? Hyperpigmentation, deficiency of pigmentation, fair hair, light skin color, yellow you are, and blue eyes. So those are the characteristics of the phenylketonuria, right? Okay. Now we'll talk more about the phenylketonuria.
hmm,
Now, causes deficiency of phenylalanine hydroxylase. So that is important. Now even you remember that in the video, I'll send the video, so that is that synthesis of BH4, right? So next one, tetrahydro. So what is that BH4? Huh? BH4. Can you find it and tell me what is BH4? It is in the video. BH4.
Tetrahydro bio
Biopterine.
biopterine.
Tetrahydrobiopterine. So BH4. Right. So those things sometimes we never know. No? Right. Okay. Characteristics of the classical PKU. Classical PKU is an inherited disease due to 100 or more different mutations. Phenylalanine hydroxylase gene. Elevated phenylalanine. So phenyl lactate, phenyl acetate, uh, phenyl acetate. So phenyl as what are these two terms? Are they same?
No, phenyl lactate and phenyl acetate.
All right. Phenyl lactate and phenyl acetate and phenyl pyruvate give urine characteristics, mousy odor. Right. CNS. CNS means central nervous symptoms. Mental retardation and the failure to grow. Failure to walk or talk. Seizures, microcephaly. Okay, right? Search about microcephaly. What it is. Microcephaly. So I told that it is, I'm going to ask it. Next one is a hypopigmentation, deficiency of the pigmentation, fair hair, light skin and color and blue eyes, right? Next one is a homocystinuria, defect of metabolism of homocysteine, high plasma and urine level of homocysteine and methionine, low level of cysteine, autosomal recessive, deficiency in the cystathionine beta synthase, most common, converts homocysteine and cystathionine. Symptoms: ectopic lentis, that means displacement of lens of the eyes. Skeletal abdomen is tendency to form thrombi, blood clots, osteoporosis, and neurological defeat. Treatment: restriction of methionine intake, supplementation of B6, B12, and folate. Right. So now here, the next one is the amino acid metabolism urea cycle. So urea cycle. So here, right, so that there's a metabolism of ammonia, source of ammonia, right? So there are so many things, right? So now here I'm going to show you a small video about the urea cycle, right? So discuss everything. So this is the syllabus of the urea cycle, right? We'll watch a little clip to understand about urea cycle. So that is then it will be easy for you.